So we'll now move on to our teaching lecturer, Professor David Isenberg. We had some connection difficulties, so I'm keen to get you on now, David, while we've got you. So David is the Versus Arthritis Professor and Director of the University College London Centre of Rheumatology. He has a specialist interest in lupus, but also phospholipid syndrome, Sjögren's syndrome, and has extensive work on double-stranded DNA, phospholipid antibodies, and the tools to measure disease activity in lupus. And of course, long COVID or post-COVID, or the inflammatory tail of COVID, has borne some similarities to lupus. So we're often asked if if treatments along those approaches would be useful. Hydroxychloroquine obviously was in the press mainly last year. So I'll pass on to David, who's also been past president, British Society of Rheumatology's Systemic Lupus International Collaborating Clinics, and is a fellow of the Academy of Medical Sciences. Dear David, really pleasure to have you here.
Thank you, Michael. Can you hear me okay?
Hear you perfectly.
So many thanks. I must say, if a year ago you had told me I would be talking to some very distinguished neurological colleagues on the subject of COVID, I think I would have doubted your sanity. But it's been an amazing year for us all, an amazing journey. Could I take a look at the first slide, please? Thank you. And the next slide, please. So here's a remarkable quote from Bill Gates at the Munich Security Conference 2017, quoting, "Epidemiologists say a fast-moving airborne pathogen could kill more than 30 million people in less than a year." Next slide, please. You could say that we got off perhaps relatively lightly because, as Mike has just told us, it's now affected about 115 million people and it's killed about 2.5 million in the first year. Next slide, please. Now, of course, this pandemic is just one of a number of pandemics who have affected the human species over the past millennia. Smallpox, its origin is debated, but some believe it began in India perhaps 11,000 years ago. But there are well-described outbreaks occurring in Egypt about 5,000 years ago and in Athens over 2,000 years ago. It's known to have spread from China to Japan, and it's known that the expansion, which a number of Arab armies led to its spread in North Africa, Spain, and Portugal, and it's known that the Crusades led to it being spread all over Europe. Next slide, please. So what's fascinating to me, actually, and some of you perhaps know this story, is that actually it was smallpox which led to the whole question, the whole issue of vaccination. And Jenner was a West Country general practitioner, and he made this very simple clinical observation that milkmaids who got cowpox didn't get smallpox. And he took material from a cowpox sore in a woman called Sarah Neems, and he inoculated this 9-year-old boy called James Phipps, the son of his gardener, and months later exposed him to patients with smallpox. As I've said, this probably wouldn't have passed the local ethics committee, that's what he did, and James Phipps remained well. But of course, it was the development of the smallpox vaccine which led to an intense eradication program which began in 1962, and the last naturally occurring case occurred in Somalia in 1977. But tragically, the last recorded case was actually a medical photographer at the University of Birmingham who worked on the floor above the microbiology department where they were doing some smallpox research. And clearly the virus got out and she died tragically of smallpox in 1978. And she's the last known person on the planet to have died of smallpox. Next slide, please. The Spanish flu is interesting because, as most of you know, it has nothing very much to do with Spain. It has a lot to do with the fact that the Spanish press was very free, and rather freer than the British press was, and the American press even, at the end of the First World War. And most of the stories about the Spanish flu actually came out via the Spanish press. It seems to have originated in Kansas, so one of the very few things to come out of Kansas that was good, I suppose. And it was brought by the American service personnel to army camps at the end of the First World War. And it seems to have spread very rapidly to other people in those camps who then were about to go home, and they took it all home with them. They spread it all around the world. And intriguingly, there were 3 main waves of this epidemic, and the second one was the big one. Occurred in October 1918. It killed 50 million people. And even more remarkably, the virus was recovered and its sequence worked out from tissue taken from a body of an Inuit woman called Lucy, buried in the Canadian permafrost. This was in 2002. Really quite an amazing story. And of course, as I'm sure you know, there was no vaccine ever developed against the Spanish flu. It simply died out naturally. Next slide, please. And we can't, of course, forget the HIV pandemic due to a lentivirus, thought possibly to have originated in West Africa. That's still a little bit uncertain. It killed approximately 32 million people. No vaccine has ever been developed, but we have, of course, developed very successful antiviral drugs. Next slide, please. So here are some interesting comparisons between SARS-CoV-2, the original SARS-CoV, and the pandemic of 1918. And it's particularly striking that symptom onset to maximum infectivity with SARS-CoV-2 is very, very rapid. But the proportion with mild cases is actually still quite high in SARS-CoV-2, as indeed it was with the pandemic influenza of 1918. Next slide, please. Now, just to remind you, some of you will know this perhaps, there are a number of different types of coronaviruses which were first characterized back in 1965. They're thought to be at least 4 mild subtypes, and it's believed that about 15% of common colds are actually due to these mild coronavirus subtypes, which probably explains some of the immunology I'm going to talk to you about a little bit later on. But there are 4 major subtypes, including those which caused the original SARS epidemic or pandemic in 2003 and the MERS one in 2002. And now, of course, we look at the SARS-CoV-2. We've been talking just recently, the previous speaker, about the UK/Kent variant, South African variant, the Brazilian variant, and there will undoubtedly be other variants too. Next slide, please. So what are the risk factors for COVID-2? These came to the fore very quickly with the first a study of the causes of death in North Italy, which were published just under a year ago. A study of 500 people who died from COVID in North Italy. And then, as you probably will remember, North Italy had many business connections with Wuhan. There were direct flights going to Wuhan from the northern part of Italy. And in that first study of 500 people who died, the average age of death was 78. 99.1% of those who died had at least one serious underlying disease, and 49% had 3 serious underlying diseases. So in the study that was published in the BMJ, 15,000 people, of whom nearly 4,000 had died, the key factors in determining outcome were age. Clearly over 80 is not good. Being male was not good. And having almost any kind of chronic disease, cardiac, respiratory, kidney, neurological, liver disease, these are all poor prognostic features. Obesity is a factor. Ethnicity, some of you will have seen the program, I think it was on the BBC 2 or 3 nights ago about this, and dementia too. Next slide, please. Coming from this as a rheumatologist, I, of course, am very interested in the question of whether patients with rheumatic diseases, especially the sinister ones where patients are already on steroids and immunosuppressive drugs, are those patients more susceptible? Now, interestingly, from my own practice, and as Michael has hinted at, I see literally look after hundreds of patients with lupus and equally large numbers of patients with rheumatoid arthritis. I was not struck by any vast increase in deaths due to COVID anyway, in the lupus patients. And I think the reasons for that are twofold. Number one, the lupus patients, certainly in the UK, were very, very careful to shield. They stayed home and they didn't go out. And number two, of course, they tend to be rather younger. The average age of onset of the 770 patients in our lupus cohort is 28 years. So these patients are younger and they stayed home. And I think that's probably why we've seen a small number of deaths in the lupus cohort, but not that many. But the question of immunosuppressive drugs is also very interesting, not least because several, as Michael again hinted at, several rheumatological drugs appear to be beneficial. Steroids, clearly. Tocilizumab, I'm going to tell you about in a little bit more detail. And there are some ongoing studies of baricitinib, which blocks the JAK-STAT inhibition, and adalimumab, sorry about the typo there, but not hydroxychloroquine, although that was suggested by colleagues in France about a year ago. I think it's now widely accepted that hydroxychloroquine, wonderful drug as it is not unfortunately going to help us against COVID. Next slide, please. So this is the story of the one rheumatologist ever to win the Nobel Prize. This is how steroids got invented, as it were. His name was Philip Hensch, a very remarkable guy working at the Mayo Clinic in North America. Next slide, please. So Hensch's story is that as a clinician, he observed that if you had rheumatoid arthritis and you got jaundiced, you got better, which was very curious. That was in the 1920s. In the 1930s, he observed that if you had rheumatoid arthritis and you got pregnant, you got better. And he began to surmise that maybe if the body is under some kind of stress or strain, it produces an anti-inflammatory something. That what he was. Fortunately for him, he was very friendly with the guy who was to share the Nobel Prize with him in 1950, Edward Kendall. Kendall was extremely interested in the secretions of the adrenal gland, and the two of them conjectured that the anti-rheumatic substance X, as they referred to it, might be an adrenal hormone. And towards the end of the Second World War, they took to going down to the abattoirs just outside the Mayo Clinic, and they asked for the adrenal glands to be given to them, which they were from the cattle. They took them back to the lab, they literally squeezed them, and they began to analyze the fluid. And the very first substance which they managed to purify, which they called compound X, was actually cortisone. And what they did was to treat 14 patients with severe rheumatoid arthritis, and they filmed them. And these patients with very, very severe disease were asked to walk across the old wooden gymnasium floor at the Mayo Clinic. And it's very painful. I've seen these pictures, very, very painful to watch, and they can barely do it. They've got to get over a little set of wooden steps. They then get a week's set of injections of compound X, actually cortisone, and now, hey presto, they run over the steps, magic darts sort of thing. And when the results of treating these first 14 patients were reported at medical meetings, it was said that even senior physicians were so impressed they stood on their chairs to cheer. So I naturally would hope at the end of this talk that one or two will stand on a chair, not just to change a light bulb, but to cheer. Next slide, please. So dexamethasone is the steroid for which there is the greatest evidence, and the EMA concluded after reviewing results of dexamethasone from the recovery study, and I quote, "Dexamethasone can be considered a treatment option for patients who require oxygen therapy, from supplemented oxygen to mechanical ventilation. It does seem to be effective." Next slide, please. Mike has already told us a little bit about the cytokine storm, or hinted at that, which is now widely reported in significant numbers of patients with COVID. This is associated with the overproduction of several cytokines, including IL-6, IL-1, and TNF-alpha. And of course, in rheumatology, for the past 20 years, we've been using monoclonal antibodies to precisely these targets, and they've been very successful in the treatment of rheumatoid arthritis, psoriatic arthritis, and ankylosing spondylitis. And thus, studies have been going on to determine whether these drugs might be useful in treating the cytokine storm that's associated with COVID infection. Next slide, please. And I want to focus your attention in particular on IL-6, a very significant pro-inflammatory cytokine. It's secreted by macrophages in response to specific microbial molecules. It stimulates acute phase synthesis and the production of neutrophils in the bone marrow. It supports the growth of B cells, and it's antagonistic to regulatory T cells, and it's thus thought to have a key role in inflammatory and autoimmune processes. And the key work that was done in understanding the importance of IL-6 was done by a Japanese worker called Kishimoto. Next slide, please. And you know your famous, next slide. Yes, let's just keep that picture up. Go back to that next picture, please. You know you're famous when you get a lecture series named after you. Now, Kishimoto works in Osaka in Japan. He's semi-retired now, but he and his private company developed what became tocilizumab, the first anti-IL-6 receptor monoclonal, about 25 years ago. A very small study was done in Japan. Next slide. Sorry, can you go back one slide, please? And this was the paper that colleagues and I were asked, based on the work that we were asked to do in England about 20 years ago, the first trial of tocilizumab in rheumatoid arthritis. And the next slide, and then the slide after that. Thank you. Kishimoto is seen on the bottom left down here. That's Kishimoto having a meal. I was asked to give the lecture in his lecture series about 7 or 8 years ago. He took me out to eat, which was very nice, and we had a very nice time. Very interesting guy, speaks quite good English, and really has made a huge contribution in this area. Next slide, please. And the REMAP-CAP study has shown that in terms of hospital mortality, both the IL-6 receptor blockers, tocilizumab and sarilumab, are better than the controls. And so we now reckon that tocilizumab is a useful drug to use in the treatment of severe COVID infections. Next slide, please. So we now have 6 effective vaccines, at least I should say 6. You'll know about the AstraZeneca one and the Pfizer one and the Moderna one, I'm quite sure. As you know, AstraZeneca virus has been suggested might not be as effective against the South African variant, but the Pfizer, BioNTech and the Moderna ones are are extremely effective, but the allergic reactions that we see in all of these things are relatively rare, particularly apparently in the Pfizer and the Moderna ones. Next slide, please. We also have available now the Johnson & Johnson, which is very interesting because it's a single shot, unlike the others which are double shots. They all essentially, or many of them, integrate adenovirus with genes encoding for part of the coronavirus. Novavax has the same sort of thing, and the Russian Sputnik virus is broadly similar as well. So we now have, and it is a tribute to the science, that in less than a year, actually, we've gone from having barely even hearing the word coronavirus to having 6 effective vaccines against it. Next slide, please.
You've got 5 minutes, David.
So I don't want to get too political, but it's probably worthwhile pointing out that the UK government has done something right, and that's in securing large numbers of doses of 3 of these main sorts of vaccines. Next slide, please. So why do individuals have such different immune responses to SARS-CoV-2? Many of patients who are affected by these viruses are actually asymptomatic. A number of different clusters of COVID have been identified. And as we've just been hearing about, long-term sequelae, long COVID is a particular problem with potential psychiatric sort of disorders. Next slide, please. And I mentioned that you do get the common cold, or versions of the common cold, being caused by these milder versions of COVID. Next slide, please. And it's very interesting to reflect that young people, children actually, have been demonstrated to have been exposed to COVID infections, and might possibly be the reason, or part of the reason, why children under the age of 10 or 15 it seems that they very rarely get infected. Next slide, please. Now, in terms of the antibody immunity to the SARS-CoV-2, the spike protein is clearly central to this, and the component that I've shown you here called S2 is particularly important because many of the antibodies which are formed against COVID are formed against the S2, but it's actually the S1, the more distal aspect, which actually links to the ACE receptor, which gets the virus into. Sorry, I'm so sorry. It's running the other way. It's the S2 which connects the virus into the cells which infects. Next slide, please. And this is some work done to demonstrate the presence of very sensitive and specific assays detecting antibodies to SARS-CoV-2, including a number of cross-reactive assays. And if you look at the graph on the left-hand side over here, if you can see that, you can see quite profound antibody responses are developed against COVID-19. Next slide, please. And what's very interesting is the demonstration by George Cassiotis from the Institute of Child Health of pre-existing de novo humoral immunity against SARS-CoV-2 in human beings. So in other words, presumably because of a cross-reacting antibody, antibodies are detectable in many human beings, even if they haven't been exposed to SARS-CoV-2 by virtue of having been exposed to previously to other COVID viruses. Next slide, please. Nearly done. And this is just to show that pre-existing and de novo humoral immunity does exist. It's seen in this slide over here on the right-hand side. Next slide, please. It's mostly directed against the S2 component, and it largely consists of an IgG response. Next slide, please. And you see the presence of the antibodies to SARS-CoV-2 can be identified in relatively young people. Next slide, please. And so in conclusion, the COVID-19 pandemic is, I've said, merely the latest pandemic to affect human beings. It's clearly highly infectious, but not as lethal as some previous infections, such as smallpox. As I've said, remarkably, within a year of its emergence, several highly effective vaccines have been produced. And fascinatingly, details of its immunological consequences are being studied, but it does appear that some people actually have antibodies to the S2 protein, even though they have not been infected with SARS-CoV-2 by virtue of having been exposed previously to other sorts of COVID vaccines. Particular thanks to Claire Deakin for helping me to put some of these slides together. And final slide, please, and to a group of other people who've been involved in some of the work I was showing you at the end there. Michael, thanks very much for your invitation.
Thanks, David. Thanks for a tour de force. And real insights. I think we've always in neurology had many of our insights for therapeutic immunology from rheumatology, and hung on the coattails of what's being done in lupus and vasculitis. So it seems, I'll ask one question, but everyone do please ask a question or two, and if at 1:30 in a couple of minutes, we should start with one Data Blitz talk and then Dr. Nordvid's talk and then the final Data Blitz. I mean, one question I'd like to ask, because it seems that there are people who have bad COVID at the very beginning, and they're not necessarily the people who get long COVID. And, do you think they're two different diseases? And what are clearly dexamethasone and tocilizumab are telling us something about the mechanisms of organ injury or long-standing symptoms from COVID, if you had to choose one or two surrogate markers in trials to see if a treatment worked, because you don't have in long COVID a hard endpoint like death or ITU admission, what would you choose?
I think the truth is, Michael, that the question is a perfectly good one, but I think it's unanswerable. I think we need to know far more, especially about the factors which lead to long COVID. I think it's now pretty clear what predisposes to a bad outcome with in a more acute setting is I think that's pretty clear now. But what determines that you're going to get long COVID, I don't think that's at all clear. And I think that remains to be determined. It is possible that certain cytokines may linger rather longer, possibly IL-6 one, but the truth is we just don't know.
Yeah, thanks so much. Thanks again, and do please stick around for the chat and Professor Compston's lecture at 8. Thanks, David.